Hi everyone. So this photo is taken at Tocal Agricultural College, and that's two of my honours students. So you've seen some of the things I'm covering in this talk, both in Liz's talk and also in Tom's. So Plan B has been created to help secure the Australian beekeeping industry, and we have support not only from beekeeping industry but also from the industries that are dependent upon them for pollination. Okay, so basically in a nutshell, the aim of Plan B is to produce happy, healthy, productive bees. So producing lots of honey but having low disease and pests, which will reduce costs, and that will foster a strong beekeeping industry and help secure pollination for those industries that are dependent upon them for pollination services. So when you're starting your breeding program, it's good to know what you're starting with. And so I've been looking at the population genetics, and I got samples from between 2 and 78 colonies from 31 different beekeepers, and I used 9 microsatellite loci to look at the population genetics within those breeders. And I did want to do genotyping by sequencing, but unfortunately, when you send bees in the post, they get very hot and you don't get good enough quality DNA to be able to do that. So hence I did it with the microsatellites. So then I used principal components analysis, which puts that genetic diversity into 2 dimensions. And there's a couple of things that I'd like to pull out here. So here, population 12 and 13, they're from 2 island populations. So it's not unexpected that they would be more distant to the rest of them. And then the other thing you'll notice is that the Western Australian, the X's, tend to be on this half of the graph. And, you know, now we know that WA considers itself quite separate to Australia a lot of the time, but this has been going on for some time with bees. And that is because they closed their borders to bees in 1977 because in the eastern states, they got European foul brood. And then there was some concern that from closing their borders that they would have an inbred population very quickly. So they implemented a breeding program in 1979. And then that was eventually handed over to beekeepers, and it's been ongoing ever since. So for some of this time, it's been maintained via artificial insemination, like Liz is doing with lots of drones. But for most of the time, what they do is they take rather a small number of colonies, so their best colonies each year, onto Rottnest Island where there's no feral bee population there. So they go there for less than 2 weeks and do all their mating there. And so because this population has been isolated for so long, back in 2008, I was looking to see if they had a problem with inbreeding, and the heterozygosity was 0.61, which is quite healthy. And so we were not too worried at that stage, but, you know, that was 13 years ago now. And so we took this opportunity with the Plan B to have another look at that population. So in 2020, they took 58 colonies to Rottnest Island for mating. And this picture here is, instead of showing each beekeeper as 1 point, we've got each individual colony as 1 point. So all the black circles are the rest of the states. And again, Western Australia has the crosses. So again, they're all on the right-hand side. This is actually a different graph with different scale, but it just happened to all turn out on the right-hand side again, which was fortunate in some ways and not in others. And so the heterozygosity again was 0.62, so almost the same as what it was back in 2008. And this is probably actually an underestimate, because what I've done here, instead of using 1 worker per colony, I took 6 drones per colony. And that's because we can't sample the queen herself, because she's a very valuable individual, whereas unfortunately, or fortunately, the males are not considered very valuable at all, and no one really cares if you kill 6 of them. And when we do that, we might miss some of the queen alleles. So this might be an underestimate of heterozygosity. And I looked at the inbreeding index, the FIS, and this here is probably an overestimation because of that missing some of the queen alleles possibly. So that's actually, it's fairly low, and I'm not concerned about that. So genetically, the Western Australian population is nice and healthy. Okay, and then as part of this program, we've also got a breeding population at New South Wales DPI. And this population was established by taking queens from basically all over the nation. So unsurprisingly, you can see that that genetic diversity is reflected here in this figure, with the pink triangles being all over the graph. So that indicates that we've got good genetic diversity to start with. The heterozygosity was also good, 0.68. The inbreeding coefficient, the FIS, is 0.18, which is a little bit high, but I think this is not really quite a valid test here, because this was taken before we started breeding this population. So there was population subdivision in there, because it was done before Liz started crossing the different lines. So you've already heard a bit about the complementary sex determination locus, but I'll just quickly go through it again in case you weren't there or you were asleep. Sorry, Tom. So in honeybees, this is the means for sex determination. So an unfertilized egg results in a drone, and a fertilized egg results in a worker. So the drone will have 1 copy of the CSD, whereas a worker will have 2 copies, and as long as they're functionally different, then all is well and good. However, if you have a fertilized egg where both copies of the CSD are not functionally different from each other, you'll get a drone rather than a worker, and then the workers will eat them before they develop very far. So this is really bad for population, well, for the colony when it happens, because they tend to fail to thrive. If the queen had only mated with 1 male, then half of her offspring would be inviable, and that results in lower honey production and just a failure to thrive, basically. So I looked at CSD diversity both in Western Australia, and here it says New South Wales, but this is not the New South Wales DPI population. This is, in fact, just another New South Wales bee breeder. COVID stopped us from being able to do the DPI ones. So this network shows you a few things. First off, I'll note that it doesn't include every site. So it doesn't include all the insertions and deletions, which there are a lot of. And you can see that there's very few alleles that are in both populations. So they tend to have different alleles, but the branches are not segregating by color. So that means while they have different alleles, the alleles that they have are very similar. And so the New South Wales beekeeper, it was just 1 beekeeper, had 32 different alleles, and Western Australia had 28, and that was from 11 different beekeepers. But obviously, that was only being about 3 or so colonies per beekeeper. So by no means have we got every single allele that are in these populations. But that number is really good. For example, in New Zealand, in 1 population, they only had 16 alleles, whereas in another study in Poland, they had about 80. So I think this is a good number. So I'd like to thank everybody that's been involved in getting this project to this point, particularly Liz. And if you'd like any resources to follow up on the Plan B program, there's the website where there's manual and different workshop recordings available there. And we're also active on the social media sites. Thank you.
Great. Thanks very much, Nadine. Very nicely presented. Interesting program. Now, do we have any questions for Nadine? So Nadine, you know how the WABs have been separate from everyone else for quite a while. Do they show any characteristics that are different from the other ones, or are they pretty much the same at the phenotypic level?
I guess at the phenotypic level, no. Maybe because they've been selecting for the same traits, maybe they might express different traits at different levels. But what I forgot to say actually is, although other bees can't go into Western Australia, the Western Australians can sell bees to the eastern states. So there is some flow that way. And sorry, I forgot to turn my camera on until just now.
That's okay. Okay, anyone else have a question or comment for Nadine? Okay, we've got a couple of minutes. So how long is the Plan B program going to run for, Nadine?
At the moment until mid-2023, but of course we're hoping we'll be around for the long haul. Yeah, so we'll be, I think by then it will feel like we're barely started. You know, there's a lot of work that goes into getting everything ready to go for these programs. You know, creating manuals, getting databases happening, and then you've got to get the beekeepers starting to collect data.
And so you've got good engagement from the beekeepers, they're all interested in being involved?
Yes, I guess the problem is getting them to actually keep records. So beekeepers aren't really known for wanting to be on computers. So we've got a few different methods for trying to get them involved. So for example, if they just write them down, at the moment we're willing to input that data for them. But yeah, I guess their time is very precious, and unless they can actually charge more for the bees that they're selling, then they can't justify that extra time input.
Okay, so you can't make them a nice easy app just to...
There is an app that has been, is being developed and is being tested, but I have not experienced it myself.
Very good. Okay, well, last